bamboo coral?
The skeletons have these nodes, which are in between these calcite part of the axes, and it looks like the the bamboo nodes that you see. And so it's hard to see from this view, but if you look closely at the axis and at the skeletons when there's no fleshy material on it, you can really see those nodes.
Thank you.
That's a good look at that. Thanks. And then there's just below that and a little to the left is a black coral. There's two down there actually. And I think this is probably just similar to what they collected, but let's take a zoom on that. And then let's get a nice still of that. And then there was one to the left of it that looked a little bigger.
So it's just a rate limiting factor.
So there's 3 black corals here kind of all in a row. I'm interested in looking in that middle one.
Mm-hmm. Okay, zooming in. Coming wide.
Okay, I guess that's good. Kind of hard to see straight on like that because it's oriented so vertically.
I could come around to the side if you want.
Okay, that'd be nice. Thanks.
Sorry, I had to bring the expert up here to
coming wide.
Get some advice. Thank you, Greg.
Zooming in. There we go.
That's a good look at that. I think that's similar to what we've just collected. So we can zoom out and move along.
All right, full wide.
Copy, moving along.
Boy, this is a really neat feature, especially after traversing that mud habitat for a while. It looks especially spectacular.
Indeed. Can you, Jenny, remind us how these rock features are formed, or tell us anything about the rock that we're seeing?
I'll try. So we are west of the continental shelf, and there's been a lot of uplifting.
Can I get a top of the screen, come up a little bit?
I see it, yeah.
There's a coral.
There
we go. Let's take a look at that.
Now I know what you're looking for.
This region is a tectonically active area, and there's been a bit of uplifting and
You're in the light bar.
Yes.
lifting of different layers of the ocean. And there's just
wait.
these troughs at the end of the shelf there that dip down into the abyss. Guy describes it best, but basically over time, if things have gotten pushed
Okay,
up
can
and
we
vaulted,
zoom out? I was actually interested in the white coral that was next to it.
we get some of these
And if I could,
deeper features.
it's just off the bottom right corner of the screen now.
So I didn't know what you wanted. Just to the right of the lasers now?
Just to the right of the lasers, you got it.
Gelatinous creature in the way. Yep. Should be a full zoom there. I'm parked on the, as you can see by the raining stuff.
That's great. Let's get some nice shots of that. I'm not sure what that is.
So that's all you want, is it?
Yeah, would
Or
it
you
be
wanna look
possible
at
to
it?
come around to the left side and see
Yeah, can
that
do.
side of it?
Okay, Wayne, you can zoom in there.
Zooming in.
Might be a little close there, but
That's nice. If we just hang here for a second. Caitlin and Gary, I don't know what this is. Do you guys have any ideas?
okay, Wayne, go for full zoom there.
All right. Full zoom. Oh. Let's see if I can get it.
I vaguely remember seeing this
Try one more. There, you should
in
be able to
Vidake
push all the way in
2
there.
years
mate.
ago. Remember we called it something with bells on it?
Oh,
Oh, there's a
But I
tube
don't
worm
remember the
there.
name. Norella?
yeah, I was wondering if it was that. That one looks so fluffy.
That porch light wasn't there yesterday.
Greg fixed it. And we also tilted the other lights down a little bit to try and alleviate that shadow between the optimal spot and the porch.
It might be Norella, as Jenny was just pointing out. So, okay, we got some nice shots of it, so we can zoom out and move on.
Okay.
It's pretty.
Roger that.
It is very pretty.
I could look at that all day.
Tube
Zooming
worms
in.
out too.
Yeah.
Full wide.
Roger. Just gonna center up my camera here again. It's on whacked.
Lounge, I didn't catch you if you were talking. Hi there. We said maybe Norella? Oh, Paris now. That's on our list. Should we collect it?
When in doubt, put it in the mailbox.
Okay. All right. We can just keep moving along this wall and look for some more corals and sponges.
Moving along.
Oh, there's a nice maybe Caligorgia. The bottom of the screen. We're going to zoom on that.
Sure. Okay, Wayne. Do you want a side view of it?
Side view would be great.
Can do.
All right, standing by.
Oh, you can zoom in a little bit there.
Okay.
That's good.
Photo bum.
Looked like it was in slow motion.
Okay, beautiful. That was a nice shot.
Okay, coming wide. Nice.
Which one was that, Danny?
That was Caligorgia.
Okay. Primnoid, okay. Cranked up the AC, it's getting chilly in here.
When these surveys are analyzed, what are some of the metrics
Look at
that
cute little guy.
Caitlin and others are looking for? Species richness, diversity, what are some of the indicators of a healthy habitat?
Well, things we're looking at, sometimes we're just looking to see what species are there as a species inventory. How many species do we have? And sometimes we're looking to see how the area that we're looking at, how many species do we have in that area, so we can get density estimates. Sometimes we are looking at richness or diversity.
Habitat type.
Habitat type and counts of organisms. Mainly because we've been exploring some of these deep habitats for the first time. We're just trying to figure out what's there.
Sure.
But with some of these quantitative transects that we've been doing with more prescribed directions, we'll be looking at trying to get density. So we know the area that's surveyed, and then we count the organisms in that area.
I've put down sediment tiles before and looked to see what organisms colonized it. And that was a You know, 8-inch by 8-inch space, so I can't imagine doing such a task for such a wide area.
Yeah.
Well, the anemones like it here. They're pretty cozy.
Sure do. I see that little white coral that's just by the lasers,
Yes.
just above the lasers.
Want to zoom in a bit there, Wayne?
Sure. Zooming in.
Perfect.
Nice little guy. Okay. All right, and zoom out.
Coming wide. Full wide.
Thank you.
Viewers at home might notice all this white snow drifting by, and that's marine snow, which drifts down from the surface waters to this habitat and is food for these corals and some of the serpent stars and other animals that are cast open wide, the crinoids. It's basically stuff that has died and decayed. Could be some zooplankton in there too, drifting by. With the occasional ctenophore cloud.
Let's take a peek at that crab.
Sure.
Great, we can zoom out.
Zooming out. Full wide.
So this is a great feature, and it's extending a little farther than I initially thought, so we should be able to follow this around. It looks like the contour curves around and will bring us up to our next point. Be great if we would have this interesting habitat to follow along that contour.
One viewer's curious about the green coating or brownish green coating on the hard substrate. Can you describe what that is?
I don't know what that is. Sometimes I think it's the color of the rock, and it could be like a bacterial mat or something. I'm not sure exactly what that is.
You're seeing a lot of mud on top of the rock. We call it mud-draped rock.
Oh.
But I've been noticing that hue too, and it seems to change as we get closer with the lights and get different hues.
Mm-hmm.
Could be a molt or...
Someone had crab for dinner.
Yeah.
Beautiful ledge.
still want to pry a rock off of it. Is this speed okay for you guys?
Yeah, this is good. The speed is good. This is a nice distance from the wall that allows us to see what's on the wall, but not be so close we can't see what's coming too. So that's one thing we're looking for. And then we're moving at a nice speed, which keeps us moving, but it still allows us to stop and zoom in on things. So this is good for this kind of exploratory mode that we're in right now.
So many anemones. Can you, excuse me, can you tell us a little bit more about them?
Anemones are cnidarians, so they're related to corals. They're capturing food in their tentacles, so they are benefiting from all this marine snow that's falling down. I don't know too many factoids about anemones, but I'm sure we can look some up and talk about them.
I think that orangey one that we just passed, that big one, is called Stomphia?
Stomphia could be.
Is that it? I don't know what these purple ones are, though, that are so feathery.
These might be corallomorphs. There's a few different species of corallomorphs that we've been seeing.
Anemones in shallow waters are a little bit more predatory and eat bigger things like crabs and snails that might land in them. I even saw a picture of a Urticina piscivora, which is a fish-eating anemone in the kelp forest that got a bird, a common murre, dove right into it.
Oh my gosh.
That's wild.
I don't know who won in the end there,
I
but it
know.
was really like, whoa.
A bit more than a bargain for maybe.
You interested in a partial zoom on this interesting corner?
Yeah, sure. That'd be great.
Let's see.
We've got a nice Parastinella here. We've seen a few of those on the earlier dive, and they often grow in this kind of cup-shaped formation, and they tend to have a lot of brittle stars and serpent stars, and this one even has a nice crinoid.
Yeah, these corals specifically show just what great habitat corals are for other animals that we often call associates. So animals that live on and around to help them feed on the marine snow and help them get the nutrients they need to thrive.
Looks like one of those anemones is all closed up there on the rock. That's kind of a cool pattern. They look like big baskets. These corals. These feather stars are actually a type of echinoderm as well. They're ancient. Been around for a long time. Still living, and every once in a while we'll see them swimming through the water column.
Okay, full wide.
Can we zoom in on that kind of top
Cluster.
part of the rock there?
Anything in particular?
Nice.
or
That's
just
it.
the whole thing.
Yeah, you're great. I thought I had seen something, but it was not what I thought it was. So you can zoom out. Thank
Okay,
you.
coming wide. Full wide.
You can zoom in on that bamboo coral when you get there.
Copy that.
Okay, Wayne.
Nice, thank you.
Okay. Zoom, coming wide.
Roger.
Full wide. Oh, wow. Ready for a zoom?
Sure.
Okay, coming wide.
So I am happy for us to stay in this contour between these two white contour lines as we just move around this feature here.
I could stay here forever.
There's another crab.
It's 2 degrees Celsius here. It's chilly.
You don't realize how cold that is till you stick your hand in that bio box.
And that's probably warmed up maybe a degree.
All right, we can move along.
Moving on.
For folks who have joined us, we are about 30 miles off the coast of the northern Sonoma coast of the Walala area, south of Point Arena, and just south of the northern boundary of the Greater Farallones National Marine Sanctuary. And we are on the western edge of the continental slope in some deep features, exploring and doing some transects in between too. We're in exploring mode right now. In California, in case you're tuning in from somewhere else.
This is another Bathypathes black coral on the top of the screen, and then 2 other black corals below it. I'm not sure what those are. We've collected something similar just a little bit ago.
Would you like to see them closer?
That looks good, actually.
Someone tuning in was curious about El Niños and how that impacts deep ocean and corals specifically.
Well, as we've talked about, the deep ocean tends to stay quite constant in temperature, but it can be affected by surface waters in that the food raining down to the deep ocean comes from the surface. So things that affect the surface of the ocean can affect the deep sea.
There you are.
Oh, there's a little octopus. One of the things we are studying down here that isn't El Nino related, but carbon dioxide related, is studying carbonate chemistry. And we've been taking some water samples near corals to send back to be analyzed to find out what the aragonite saturation is, which is used to understand the levels of carbonates in the water. And those carbonates are needed for animals that build their skeletons out of seawater, including elements like carbonate. And carbonates are getting harder to come by as we increase carbon dioxide with industrialization and fossil fuel burning and deforestation, because the ocean has been absorbing this carbon dioxide. And it's, in some places, the ocean shifting rather rapidly. We don't know too much about the deep sea in terms of shifting down here, so taking these water samples is really helpful to start to get a picture of what's happening down here. The most important thing we can do is to reduce that input of carbon dioxide by finding ways to create energy that don't make carbon dioxide.
Okay, I'm going to zoom in a bit. Wide.
Yeah, I'm touching somewhere. Not sure where.
Okay.
Fortunately.
Another viewer had a question about at what point does the seafloor no longer, or at what point is it no longer considered California and has another designation?
So state waters extend out to 3 miles, but I think, yeah, Jan, go ahead.
Cover it.
Of Parastanella? Okay. If you want it, we'll get it. No, we're exploring. Okay, and can we just get a small clip? Okay. So, we want to get a collection of Parastanella. Jan wants that. So those are kind of the large coral that we were just on, the one with all the brittle stars on it,
The
just
bowl
up
shape?
above. Yeah, bowl shaped.
Okay.
And we can just get a a small clip of that. We can do that without damaging the coral.
So our state waters, state of California, go out 3 nautical miles, and then it becomes federal waters out to 200 nautical miles, where we call that the exclusive economic zone.
Thank you.
So we're in federal waters right now.
Does the fact that we're part of a designated reserve or park change whether it's a part of the state or not?
No, we're still in federal waters. The only thing that's different is how fisheries are managed. And the state of California manages fisheries in collaboration with the Pacific Fisheries Management Council, which manages fisheries for the whole West Coast. So in California, they consult on species in California. And so in some cases, they set regulations that pertain to state waters and federal waters.
Pulls in there, wait.
Okay, zooming in.
The Greater Farallones National Marine Sanctuary is in both state waters and in federal waters. Cordell Bank National Marine Sanctuary is entirely in federal waters. So Greater Farallones National Marine Sanctuary coordinates a lot with the state and the state waters, and also they come up to, I think it's, they come up to the shoreline as well. I can't remember if that's mean high tide or exactly what the designation is, but they overlap with some land, national parks, national seashore, and other designations. So there's a lot of coordination.
While I get them in about.
And then if you want to get a little confusing, we have a network of state marine protected areas that were designated by the state of California with different regulations. So it's good to brush up on the marine protected area terminology, because you could say marine protected area and it can mean several different things. That might be a blue water conversation. There's a lot more time.
Okay, coming wide.
Roger, thank you.
This thing's gorgeous.
So just
All right, so what, where do you think you can get here, Dan?
I think, here I'll poke before I cut the, kind of the bit hanging out here maybe.
Okay.
Or I can try and get in and get a branch there. Or I could take the ear right here.
Oh, that might be good, an easy place to get. Kind of maybe in where you see a little indentation and a little red guy there, if you kind of put the bottom claw there.
Yeah,
Okay, great.
sampling often takes the entire control team support to have it be successful. So our data logger is telling them what number sample and where to place the sample. Our scientist is advising on what to sample. Our ROV pilots are obviously manipulating the arms and having the ROVs in position with the support of the navigator. And the video engineer is helping us to clearly see the specimen for a successful snip.
Okay, Wayne, you can zoom in there if you want.
Yeah, zooming in.
Looks like there might be a little associate on there.
I think we got
Little
the
friend.
shrimp.
Is that enough for you?
Yes, that'll work.
Maybe a benthic ctenophore on there. Something. So one of the reasons we collect snippets here and there and samples is to positively identify them. You can't do that just by looking at them or by photographing them. You actually have to put them under a microscope and look at the skeletal structure of their body. And the sclerites are a part of their skeletal structure that really uniquely identify them. So Gary Williams, who's on board with us from the California Academy of Sciences, will be helping identify all the samples we collect on this expedition.
Coming wide.
Roger, thank you. You guys okay with...
The coral will regenerate. That was a nice small snippet.
It was very tidy.
Very
you guys
tidy.
okay with one of the small bio boxes on the starboard side?
Yeah, we can go ahead and put that in starboard box A. That's empty.
I believe it is.
Taylor Ann, I have sample 026.
Yep, that is correct.
Roger. Do we have other floaty things in that bio box?
I don't think there's
Nope,
anything.
there's nothing else in there.
Ooh. Open
Oh,
house.
yeah, I forgot to ask. Is this a floater or a sinker?
It's antipitherian, so it has the potential to be a floater.
It's an anti-what?
Black coral.
Black coral.
It's a primnoid.
Is it? See, this is what happens when a geographer starts talking.
You went to Salvo. Oh, I can do it. Sorry. Ah,
I gotcha.
camera. That one. That's the one I wanted. Thank you.
And I'm gonna. You got that one?
Yeah.
Antonella?
Thruster is secure.
Yeah, you can turn them off. They should.
I do lateral as well.
Laterals? Okay. Good. Okay, your lateral thrusters and starboard vertical are secure.
Thank you.
And we are still on the seafloor.
Yeah, I just got ahead on there to hold us.
Do we always land before sampling?
Not always. We do samples on the fly. But typically we land because we need the stability to operate the manipulator from a stable platform. I think I'm hung on I get hung on the light bar out there. Cannot go down.
Can't tell.
That's what we wanted. The manipulator touches the light bar out there, and then it won't go into the box.
And for viewers watching the quad screen, you can see that manipulator arm placing the sample into the collection box. Where we will retrieve it and process it in the EV Nautilus's wet lab.
There's not enough room in there to really open the jaws very far. Did he come out?
Thought I saw it drop.
Looks like it did.
It went in and didn't come out, so.
Yay, finally.
Yay, good job.
Well done.
Okay, and
Thrusters enabled.
I was thinking about doing a Niskin, but I think we'll pass on this one. We've got 2 Niskins already, and we have 3 empty bottles left, so, and we're still early in our dive, so I think I'll save some for as we go along to different depth zones.
Okay.
Copy that. Can't blame Jess on that mud. That was all me.
Thanks, Miles.
Taylor Ann, can you, as we sit a little stationary, can you tell us a little bit also about the data logging process?
Yeah, so my role as a data logger is to observe the footage from the ROV and to take down any scientific observations of species that we see, what the sediment looks like, how it changes over time, and also at the same time taking captures of those things, in addition to logging the samples that we collect and knowing where they are placed on the ROV so that we can retrieve them and label them properly. It's pretty fun. It's a lot to pay attention to at once, but you get the hang of it after a while.
You're doing a great job.
Thank you.
And then Navigator, it seems like you, Miles, repeat the location of the samples. Are you the final logger, or what's working between you two when you're communicating?
It's a geotagger.
Yeah, it's more of a geotagger, and we want our sample numbers to be the same.
Okay.
So it's sort of a, I feel I reference the data loggers more for the sample numbers. They rarely ask me for the sample number. It's good to sort of keep everybody on the same page, especially with 3 watches, you know, spinning in and out. So one watch could sample 10 samples, one watch could do 0, and then we come back on watch and have to know what the next sample number is.
Sure. So thanks everyone who's been tuning in to nautiluslive.org. It's great to receive your questions and have
Do you want to look at that black coral we just passed?
Sure. Oh.
I don't think so. I think it was too big, but you want to go look at it? Okay, we want to look at something we just passed back a few meters. There was it caught my eye, but then it kind of got blown out by the dust cloud. So back where that was.
Was it the orange one?
Yes.
Back to the scene of the crime.
It's a black coral. It's similar in shape to what we've seen before, but it's bigger.
Must have been below me there when I hit the...
I think it's down and to the left. A little. Maybe.
There was a black coral. We'll find it.
Ah.
Oh, was it that? That's a Bathypathes.
It's here somewhere in this dust cloud.
I thought I saw something different though. That's not what I was looking for.
Okay. Full wide.
Yeah.
Yeah, I didn't think we had moved that far.
Yeah, I think we might have.
Oh, this was the scene of the crime here.
Oh, okay.
Dust cloud.
Let's go back down, I guess, the same way.
Yeah, there's the octopus.
Yeah.
Yeah.
It's down to my left, you think?
I believe so, yeah.
Roger.
Let's get some more light on the action.
And thanks to all our viewers tuning in. We have people from all over the world listening in. Australia,
I think
Singapore,
we're down lower than I was before.
Yeah.
Philippines, Korea, Greece, Denmark, Germany, Canada, United Kingdom, United
You want
States.
to go back again?
I don't know. I don't think this is where it was, so we must have passed it again.
No, I'm down lower than I was before, I think.
I think so.
Yeah, we were at 1707 and 1708 depth.
I can come up again.
Welcome to everyone listening from around the world. We're happy to have you here in Greater Farallones National Marine Sanctuary off the coast of California.
You think that's what she wants?
Okay, well, if that's it, that's not what I was looking for, so we can move on because it's it's all right.
Yeah, it's my dust cloud again. It's real soft right there. We're real fortunate to have the current coming from left to right. Except for, so it's creating a bit of dust to our right.
There's the sample, just off the top of the screen.
Where we sample?
Mm-hmm.
Okay, now I know where I am. Thank you. So I came down, was a little close, hit the shelf. There it is.
Yeah, that's it. Okay, let's take a look. A side view would be great.
Copy that. Slide gently in. Okay, wait.
Let's just hang here for a second. Let's take a look at it. It's interesting. It's does that look different than what you guys collected before, Caitlin and Gary? I know, it does look kind of similar. And it looks similar to what we've been seeing, but it is a little bit bigger. But I don't know if it's different enough. Okay. Thanks for the input from the lounge. If we got some, let's get some good shots of that if we haven't already, and then we can move on. Okay, that looks great. We can zoom out and continue along this contour. So it looks like the, just want to check in with the pilot and nav. Looks like the bow of the ship is pointed in the direction that we want to go along this contour. Is that what you guys are, where you're headed as well?
Yeah.
Okay, perfect. What is that thing just up ahead?
Kind of looks like a bone. To the right of that ledge.
On the top of the ledge.
Directly above the lasers.
The second ledge, the ledge on the left. Oh, it's a blob sculpin.
Looks like a fish to me.
What does it have, his tail flopped over on itself or what?
It looked like a bone on the other side.
Gosh.
That is a blob
Oh, yeah,
sculpin, all right. Look how blobby he is. It's got a full belly.
Does have a full belly.
Maybe he ate that crab we saw.
What are you eating?
Coming wide.
Roger.
Full wide.
Wow. That fish is pretty big.
Yeah.
We do have some questions from viewers about fishing practices in this area and some of the impacts of those commercial fishing practices.
Okay, so there, the area that we're in right now, I believe, is not part of some of the changes that are being proposed, but we will be exploring some habitat, some areas that are part of essential fish habitat, which is an area that they don't allow some types of fishing. So, and then there has been a proposal recently that is going through a process of Pacific Fisheries Management Council to open some areas and close some areas. And some of the areas here are proposed to be open, not this area that we're in right now, but some of the other areas that will be surveying possibly on this dive or the next dive. And so those changes are periodically periodically reviewed, and they have been areas have been closed for some time. Some of those areas are have been proposed to be open, and some will be closed. And so we'll be surveying some of those areas later. So the impacts, certain types of fishing practices are more detrimental to the seafloor than others. And so if there is any trawling, like ground fish trawling allowed, then that